Okay, I guess we are the first animal presentation for today, and also we're really focused on RNA. In fact, the organism that I will tell you about today doesn't have any DNA methylation of any kind. And RNA is really important to everything we do in my lab, and we got to this to some very old ideas that stem from very close by to here. And it's all to do with RNA. It's a very exciting molecule when you think about gene regulation, because you can make a regulator of an RNA sequence by just providing another RNA sequence that might base pair with it through Watson-Crick base pairing. In fact, these ideas are very old, and in this particular model, the idea was that maybe transcription factors, in fact RNA molecules, would turn out to be wrong. But we already alluded to this morning to different types of RNA-based regulation that work exactly as proposed by Charcot and Monod here in this model, both at the level of direct gene regulation, making RNA from the genome, and also how these RNAs are translated into proteins later. We study in the lab lots of different aspects of RNA biology, but most of what we are interested in involves some sort of Watson-Crick base pairing between different types of RNA molecules. But I will focus today, of course, on what we think is going on with regards to transgenerational effects in C. elegans mostly. However, I want to stress that it is very important what organism you're studying. I'll try to come back this a little later as well. And we are studying mostly animals in the lab, but we're also interested in this virus here that we discovered a few years ago together with a colleague here, Marianne Felix. And just to make the point that there are a lot of living organisms, and often there's confusion because we have a very sort of anthropocentric view and think about organisms like us. But in fact, the idea that DNA is the major or only way that information is inherited is of course wrong for all organisms, but particularly dramatic so in maybe organisms that are somewhat unlike us. In C. elegans, this story starts for me a long time ago when I was still a student and very excited to read this paper here by Fire and Mello. Discovered a process they called RNA interference, or RNAi, and what they noted is when they introduced long double-stranded RNA into C. elegans, they could get effective gene knockout or knockdown of gene expression, which got them a Nobel Prize. But of course was based on earlier work in plants, which were not rewarded, unfortunately, in the same way. But the idea is now that there's a mechanism, a very dramatic mechanism of how the environment, an RNA that you stick or in fact feeds to this worm, will change gene expression within the animal. So direct effect of the environment. We still don't know today what are all the biology underlying this sort of mechanism that's exploiting the phenomenon of RNAi. But again, only a year later, when C. elegans researchers starting to genetic screen to understand what genes underlie this mechanism of RNAi, what they noted, and this is a paper from Ronald Plasterk in the Netherlands, that genes that are required for this process of RNAi are also genes that are important for silencing transposable elements that we just heard a lot about. And in fact, this again mirrored very well, which was previously understood in plants. So RNA is not just an intermediate between the genome and making proteins. RNA has important regulatory functions. RNA has the ability to regulate other RNAs, like an RNAi, and RNA plays somehow a role in dealing with transposable elements. And I want to now fast forward to a particular type of RNA pathway that we've been studying in my lab for the last 5 or 6 years ago. It's called the piRNA pathway. The details don't really matter today, but it is related to RNAi type phenomena. It's conserved in many animals and also exists in some single-cellular organisms, but there's no direct equivalent or maybe analogous pathways in plants. This is, for those of you who care, this is an embryo of our favorite organism, C. elegans, and the nuclei of the different cells at this stage are depicted here in blue. It's just staining for the DNA. And there are two cells that are very special here. They're the primordial germ cells, the only cells that will contribute genetic material, or DNA sequence at least, to the next generation. And this is, as shown here in these green blobs, this is where this pathway is active. It's in many organisms, and definitely in C. elegans, restricted to the germline. And like what I alluded to with the RNAi pathway, this piRNA pathway, at least one of its functions is to deal with transposable elements in the germline. So because it's acting in the germline, it deals with transposable elements, it's important for fertility. In fact, in most animals, systems that have been studied, if you knock out this pathway, it becomes sterile. And therefore it has been suggested this pathway is sort of some sort of guardian of the integrity of the genome. To understand this pathway, what we did then, we did genetic screens in the lab to try to find all the genes and understand how this thing works. And I'm going to spare you all the details and give you the end result. And don't be scared, this looks very molecular. But the idea is that it's all to do with marking transcripts that come from transposable elements via this piRNA pathway. And downstream of this, we will have gene silencing. And this, as I said before, happens all in the germline. So you've got specific RNA molecules, including these piRNA molecules. They're living in the cytoplasm or just outside the nucleus. And there are some protein cofactors. And once they recognize a particular transcript as being the target of silencing, then this whole machinery will move into the nucleus. And they are just in the model of Charcot-Monod, model 1. You've got the interaction between non-coding RNAs, they're part of this piRNA pathway, and a nascent transcript that's being generated somewhere from the genome, and the outcome is silencing of this gene locus. And this involves, at least in C. elegans, what Genevieve introduced this morning, changes in chromatin structure. So some marking very close to the genetic material or the DNA. But as I said, also C. elegans doesn't have DNA methylation whatsoever, so it doesn't involve any DNA methylation. Interestingly enough, and I haven't got a picture here, but of course in many systems you imagine images of oocytes and sperm, and there's a lot of other stuff in there in addition to DNA. And C. elegans, for sure, chromatin and also some of this RNA material gets transmitted from one generation to the next through these germ cells. And the outcome of regulation by these non-coding RNA pathways leads to something very dramatic, a very dramatic heritable effect. And I've blown this up here, and you get sort of 3 types of effects. And I'm sorry this didn't come out very well, but we are considering silencing with these RNA pathways a gene within C. elegans, and the idea is that this gene makes the worm green. So, for example, you would use RNA by feeding it or sticking it into the animal. You do have an environmental change to switch this green gene off. This is not changing the DNA sequence, it's just perturbating the cell by sticking this RNA. And this is the RNAi discovery, then gene expression will indeed be specifically switched off. But in offspring of this experimental animal, the expression will come back on. This is what might you expect. However, in many cases, what we and others, including originally Fire and Mello, in the original paper noted, is that you can get these stochastic types of inheritance, where if you do an experiment on one animal and you silence effectively your gene, then some of the offspring will still remember this silencing. And if you take some of their offspring, some will remember this. And a PhD student in the lab of Ronald Plasterk has done this for 70 generations. So unless you believe in homeopathy or the memory of water, there needs to be some sort of reinforcement of this memory. And this is what we've been interested in. And then if you use this very specialized pathway, this piRNA pathway that I introduced that is expressed in the germline of many animals, you can in fact get very dramatic silencing that looks very much similar to what Vincent was just alluding to, where using now this piRNA pathway, you can silence a particular gene. And even if you remove the piRNA using genetic tricks, you've got efficient silencing of this gene in all animals for many, many generations, or at least 30 in the lab. And I have to say my nomenclature is kind of terrible, and I have to work on this. But what I mean here, there is no change in the genome, there's no change in the DNA sequence, and that's what I mean here by non-genetic, but sometimes referred to as epigenetic. Okay, and then this dramatically, of course, busts this hypothesis from Weissman that there is a strict barrier between the germline and the soma, or there's or in other words, there's a very complex interaction between the genotype and the phenotype of an organism. And I think apart from the fact that C. elegans doesn't have any DNA methylation, it's a great model animal to study what is happening here. It has a generation time of only 3 days, and it has also a small genome like plants. And this one here is summarized very quickly of what we know to be true, at least for C. elegans. First, we now, including some of the molecular pathways, how RNA, for example, that you stick in the animal or feed to the animal, gets directly transmitted from the soma to the germline and is available for transmission into the next generation. You can inherit information through RNA, both via sperm and the egg. And at least for 1 generation, it is sufficient to have cytoplasmic inheritance, as we like to call it. What I mean by this, if you want to silence particular gene lockers anywhere in the genome, you don't need this particular gene to be present to bring the silencing about. This you can again do through genetic trickery in the laboratory, but this suggests to us that RNA is really the driver of the change. However, for multi-generational inheritance of a silent state downstream of these early events, you do need chromatin. In fact, we believe there's a nuclear non-coding RNA chromatin pathway that's at the core of this, very similar to what is happening in plants. To me, one of the big questions is why sometimes this works really, really well and the silencing is very robust, and sometimes it is not so. To bring this together, and maybe this fits very well with what Vincent earlier said, the way we think about this is that our genome has genes in it, but of course there's a lot of other stuff. So in the human genome, almost half of it is just mobile elements. And things like this piRNA pathway normally deal with these mobile elements, and you can then study this or study genes proper. This is only part of the truth, because really what the genome looks like is that our normal regular genes that are beautifully regulated, part of a gene regulatory network, whatever we're envisaging, are very close and interspersed with transposable elements of bits of transposable elements. There is the potential, as Vincent was saying earlier, if a transposable element jumps next to a gene, this could have dramatic effects. This is a genetic change or a change in DNA sequence directly. This can have polar effects on neighboring genes. And what makes this most exciting is that this is somehow a little bit more fuzzy. The fact that a transposable element is close to a gene might not destroy its gene directly, but might affect its regulation. And this will go through, it will be expressed for all of these different factors and might be regulated by the environment. Does this make sense? From a theoretical point of view, I think it makes a lot of sense. And in fact, without thinking of any specific organism, and this is the work from Stan Leipla's lab, who we're collaborating with to try to figure out what is happening in the case of C. elegans. If you think about an idealized organism and think about a change in environment, you can show very clearly that inheriting information in addition to the genome can be very advantageous. And in fact, for C. elegans specifically, very old, even in my book, old work here, again from France, has claimed such behavior for C. elegans. It's a very elaborate experiment to try to adapt populations of C. elegans to living at different temperatures, which were mostly rejected by the community, but I think I would like to resurrect and reanalyze and try to repeat using modern experiments. Then for C. elegans itself, what is the evidence that changes in the environment have any transmissible effect on phenotype that is not mediated by a DNA? And there are several attempts, including I listed our own attempts, and they are sort of varying degree of strength of evidence, but this data based on nutrition, stress, longevity, immunity, and so forth. And I just want, if I have a few minutes left, tell you one example of one dramatic effect, which, if I was told this a few years ago, I would never have believed it, but I think makes sense possibly in the sort of kind of framework that I've introduced just now. C. elegans in the wild lives on rotting plant material, and it really loves the smell of alcoholic substances, So this is a nice pint of cider. And in fact, a lot of C. elegans in the wild is currently collected from orchards where there's rotting plant materials as there's apples. And we know quite a bit about the nervous system in C. elegans that find such beautiful smells. And this is because C. elegans has exactly 302 neurons. We have established a whole connectome. We know how the sense of smell talks to interneurons, although there is no computer model of the brain of C. elegans working. It's probably one of the best understood animal brains. And we challenged this animal brain then in the lab using a very simple experiment where there's sort of an open arena where we put at a starting point about 100 animals down, and then we provided 2 different smelly spots, butanone, which smells quite nice but is cancerogenic, so please don't smell it, trust me, or pure ethanol. And the animals, we asked them to choose either this smell or this smell, and then after a while we'll kill them and count how many animals end up here versus here, divided by the total number of animals in the experiment. An assay has been developed by Corey Bachman many years ago. It's called the chemotaxis index. And in fact, what also been showed many years ago, you can sort of use, some people call it associative learning. I would say this is going a bit far, but you can change this behavior, the stereotype behavior, by growing up the animals in the presence of ethanol. And the question was, is there any sort of transgenerational component to this? And here's the experiment. So we give animals food in the normal conditions, and then we change the conditions to provide ethanol as part of the food, with just a little bit of smell of ethanol while they're feeding twice a day, and we do this for several days, and then we ask, does this leave kind of a mark? And this is the data, where we're looking at this chemotaxis index. So there's a y-axis here, and if you're positive, it means you prefer butanone. And if you're negative here, it means you prefer ethanol. So as I said before, the naive animals have a strong preference for butanone. Not all of them, some of them get lost, but many animals successfully choose butanone. If you then train the animals by telling them you can get food with ethanol, then we can change this behavior. Not completely, not all of them go on to ethanol, but they have a very dramatic shift in their behavior. So these are the trained animals. And then after stopping the training, the question is what happens in subsequent generations. And to our astonishment, the children remember this, and the grandchildren remember this, and some of the great-grandchildren remember this, and then things flick back, and everything has been forgotten. This we've done over and over again now for 18 months in the lab. We've got a lab in California repeating exactly this experiment as well. Take it what you will, but this is very, very robust. And then the first question we asked, having now this paradigm in hand, is, does this have anything to do with this non-coding RNA pathways that I was evoking? And the short and, to us, quite gratifying answer is yes. So we have a mutant in the lab that has exactly the same genetic sequence as our wild-type strain here, but we have taken out this RNA pathway that I was telling you about. Details don't really matter. And these animals are pretty happy. They also can smell butanone very well, and we can still train them to prefer ethanol, but they cannot pass on this information to the next generation. So to us suggesting that we have maybe here a great paradigm to study how environmental changes, possibly in the nervous system, a sense of smell, seems ridiculous, can be passed on through generations, at least in C. elegans. And that's all I would like to say. And this is all my team, and we have some amazing collaborators which are listed up there. Thank you very much.
Yes. It's an experiment that you can repeat in a pub, more or less.
Okay. In the UK, I showed an English pint slide where I thought I'd adapt to citre, it's more accurate.
So, Eric, yes, indeed, it is fascinating that you can get this kind of transmission. So, I would like you to comment. You know, we have this example in bees of feeding the queen, and of course, this is the race at every generation, and then we have perhaps other examples where things could be transmitted for longer. So do you believe that every organism or every species somehow will have adapted its whatever set of epigenetic processes to allow or not to allow this kind of epigenetic inheritance and set limits in time to it as well.
Absolutely. I think each species has probably evolved to find a sweet spot, because even the method of inheritance is subject to evolutionary processes. So absolutely. And I think by maybe learning more about environment of a particular organism, we might be able to deduce this. But I think it's still, one would need a lot of experimental data on lots of different organisms to show this. But exactly.
So I guess it's a kind of a linked question. So in your situation now, the sudden reversion you see after 2 generations, do you have any idea how that could work? And do you know, or are you going to explore other kinds of nutritional or other signals that might be propagated further or less?
Yeah, so it seems in different paradigms, most of them involve some sort of transgene type tricks. Previously, you get different lengths for which something is inherited. This could be something very specific to gene locus, and I didn't mention this again here for this particular example, but again, I'm thinking at least myself about the genes that are involved in this inheritance. Some are also being touched by transposable elements, right? That there is, and there then maybe your particular gene or where you're sitting in the genome could either be more sensitive to this kind of stuff or not so sensitive. I don't know the answer to this yet. Overall, because C. elegans uses an amplification pathway to maintain RNA-based mechanisms, we think that this amplification has some sort of threshold built in where it falls over. Just one more comment on this maybe, that work from Andy Feyer's lab using RNAi type experiments, work from Anne Brunet's lab in a completely different paradigm. In ours, they have the same sort of cliff in the F2, F3, but I haven't got any mechanistic explanation why this might be a common cliff. But others have reported, 10 generations and then.
In the case of adaptation to alcohol, do you think that it's expression of a certain olfactive receptor that is kind of switched on and then lost?
Yeah, at the moment we don't really know anything about the mechanism underlying this, and we have a wealth of mutant animals now to test. We're also looking at changes in gene expression after performing the paradigm, and this would be certainly an attractive model, but at the moment I've got no idea what the molecular equivalent of it is. And also, to be perfectly honest, I haven't even proven that it involves the nervous system at all, because in principle the alcohol could even be sensed by the germline itself. But we're hoping that it's not the case, but it's only the beginning, a lot of work to be done, I think.
Last question.
I want to go back to Lamarck then, because if I'm not mistaken, you show here a beautiful example of environmentally induced adaptive change. I mean, this is adaptive. Now your worms can survive on this ethanol. Yeah. So are you ready to say that you have vindicated Lamarck once for all here?
Well, I would maybe say that we've vindicated some ideas that have been associated with Lamarck, but after the first lecture this morning, I wouldn't want to speak for Lamarck myself before reading up on this a little bit more. The only thing I would say is that there is still no evidence that this plays any role whatsoever in adaptation of whole populations in the wild, or even anything close to evolution. But I think if it's going to be sorted out, maybe Arabidopsis or elegans are good systems, possibly flies.
Thank you.